Ok, boa tarde, pessoal. Bem, interessante de acompanharmos um pouco desse desenvolvimento de criomicroscopia, que é uma área de interesse nova aqui no Instituto de Biofísica e que vem sendo bastante reforçada mais recentemente. Então, eu gostaria de agradecer ao professor Kilder Miranda e ao Senabio pelo convite de trazer o professor Jose-Maria Carazo, e eu passo, então, a palavra para ele para fazer a apresentação do conferencista.
Obrigado, Fábio. I will switch to English. Can't speak proper Spanish. So Professor Jose-Maria Carazo is a very close collaborator. So I was just going to say a few words about his career until he reached his group now as a biocomputing unit at the National Center for Biotechnology in Madrid. Professor Jose-Maria Carazo did his master's in Granada in physics, PhD in molecular biology in Madrid as well, where he worked also very closely with IBM. At that time, Madrid was chosen as a very strong node to do imaging processing. He actually handled one of those images done by atomic force microscopy at IBM, so scanning probe microscopy. So it's something that linked to some of our groups here. Without some groups here. And so a postdoc in New York with Johann Frank, which ended up winning the Nobel Prize for his contributions to cryo electron microscopy, especially in the part of single particle analysis and computational analysis and image processing. And more recently, he has been involved very deeply with the ERIC instructor. ERIC is the acronym for the European Research Infrastructure Consortium, where he is playing a role. And we recently got a grant approved together with this group and a group in Uruguay, Peru, us here in Brazil, Senepen, and Italy and Portugal, right? So where we proposed an organization of coordinated efforts to give training on different microscopy techniques in image processing, where we have different hubs. So this we came up with this idea of having national hubs and regional hubs where we could transfer technical expertise, training, and all these things. And Jose-Maria is our PI or coordinator of this project, which I think that will make a strong difference for our groups in Brazil. So Jose-Maria, once again, thank you very much for being here, and the stage is yours.
Well, thank you very much, Kilder. Thank you, all of you, for being here. Thank you, the institution, for hosting me. I try to manage with so many chairs. But I'm going to talk to you about something that is not connected with international or research infrastructure project. It's about the science that cryo-electron microscopy is starting to make possible in a way that may not be known in a very widely manner. It's about flexibility. I am taking for granted that you all know that cryo-EM can really obtain the 3D structure of biological macromolecules at that moment down to 1.3, 0.4 Armstrong resolution with a number of procedures that are really solid and well-founded. The point is whether we are able to also obtain dynamical information, or at least conformational information. Conformation about how a structure is moving, is adapting to others, is actually breathing, and how we would eventually be able to link this information to function. In principle, why are we thinking that this is a possibility? Well, because from very first principle, cryo-electron microscopy is not an averaging technique. I mean, it's fighting noise, signal-to-noise ratio in different manners, but in principle, it is imaging each macromolecule independently. So it doesn't work with crystals, although there is a field that is microelectron diffraction that works with crystal. it does not work with the information coming from a population, but actually it works with the information coming. Each of these structures, each of these macromolecules individually will give an independent image. And that gives us the in principle, capacity to work with each of them and find the variations, the structural variation between them that would be the macromolecular flexibility, and to do it in a totally direct manner. Of course, there are buts and there are limitations in every techniques. I'm not going to make a lot of emphasis right now, but we can discuss about that. When you prepare this very nice layer of vitreous ice, there are these interfaces, the air-water interface, and many things may happen at the air-water interface in terms of denaturation and things like that. And also, of course, you always need, when I say that we work with each independent molecule independently, it is true, but we need to get signal. And maybe sometimes you have to do averages because you don't get signal. But in principle, the idea is that population of Sao Paulo, or we have no idea of age or any other thing. We have to come out with that later on in some manner. So we don't have time information. We have a snapshot information. You have had your sample. It has been in a tube. It has been resting there or having seconds or minutes to reach whatever equilibrium it has to reach, and then at a certain point, you freeze it. And the macromolecules are in whatever state they were corresponding to that equilibrium that is there. How did we start working? Well, when it was with Joachim, we did things like that with negative staining and making classes of images and things like that. And we were able, from the classes of images, that is here, to go to more or less defined classes in 3D. But really, the breakthrough was this paper in 2007, where we use a particular method that has been very useful and very successful in other fields of science. That is the maximum likelihood in a particular manner, that is expectation maximization. So we approach this, we applied essentially this maximum likelihood expectation maximization to cryo-electron microscopy images. The difference with the work before with Joachim was that here we were working with cryo-EM images, having a very low signal-to-noise ratio. You barely can see anything by eye, while with negative stain you can see perfectly the images. At that time, we knew also the limitations of our work, and the limitations are abstracted, or this is actually a sentence from the paper. These probability calculations are based on the assumption that all images are projection of one of the k underlying 3D object. So our model of the reality was you have k stable states. There is absolutely nothing in between the states. And you know beforehand how many are these states. You have to give me k. Okay, this has two major problems. Was that you may have a stable state, of course, and others that were less stable. But binary, that really there was absolutely nothing. Well, this was, of course, some shortcoming in the modeling. And the other was that you have to provide as parameter the number of those states, the number of those structural classes, of which you had no idea. So we knew that this, of course, had to get you into a more complicated way of I'm going to present in some slides some work with SARS-CoV-2, so just to introduce very, very simply that this is the famous spike of the virus. And the point I want to make in connection with what I have just said about the K classes and all that was that, of course, since you didn't know, you had to start guessing. And once you guess this has 2 classes, then you didn't reach high resolution here, then you did another classification. So this, almost by definition, had to lead to a reasonably complex workflow of image processing, because a key parameter that was the number of classes you didn't know, so you have to guess, and you have to probably guess in an educated, hierarchical manner. What do we want to do? Well, we want to do first to present you a model that is more fitted to the data. And this will result into more stabilities on the mathematical side, and will also hopefully provide a view on this intermediate that have to exist between very stable classes. At the same time, it will be automatic, so it will not ask you to go through this complicated or potentially complicated process. So the situation now has, I mean, this is a very hot topic in cryo-electron microscopy. This is our work, but these are actually other works. And you have Cryo-Dragon, that is a very nice piece of work. You have Gaussian mixture model from Steve, from Joachim. You have also Joachim Frank. You have another manifold embedding. So there are a number of approaches, perhaps 4 or 5, from people that are very much in the field. But this is telling you that this is still an unresolved issue that is really fundamental, that we think that as important the structures are, it is also how they change. And this is what the flexibility is, what these methods are all about. So before we were asking about having 2 classes. Now, in principle, we are asking about the conformational landscape of that macromolecules. And we do expect, indeed, if they are stable classes, or you have a landscape of solution and they have stable states, to have situations like that. Well, so with this introduction, this talk is about flexibility. We are not going to impose discrete classes. We certainly want to find those subtle changes that were in principle hidden by the strict K classes, and also to simplify the analysis, to give you tools to understand what is going on, and something that actually came as an afterthought of the whole approach is, okay, if we are able to know the flexibility of each of the images, maybe we are able to undo that, and maybe we are able to get maps, 3D maps that have increased local resolution. Well, you are going to see that this is actually the case. So how have we approached, in practical terms, these conformational states? The first approach that we followed was also the more obvious. I suppose that most of you have used at some moment principal component analysis. It's a tool, you have in a space with many dimensions, and you want to reduce these dimensions to just a few that just happen to carry most of the variability in your data set. So here is a little bit more complex because our data are 2D, and we want to get the 3D PCA, principal component analysis, or principal component space, but essentially is like that. Then you are going to see other couple of approaches that along the way. Very early in the pandemic, and this was 2020, we teamed up with Jason McClellan. At the time, Jason had presented the structure of the spike. He had produced the spike with a number of stabilizing mutations that were immediately taken by pharma for making the vaccinia. So the point was, and how flexible is that spike that we are all going to get one way or another in the form of vaccine. And we found that really there was a variability, that there were 3 dimensions, and this closed and open state that were being described were by no means the single state that you had. You had quite actually a continuum of conformation, and we could actually model this conformation that were related to concerted movements of the NTD, N-terminal domain, and the receptor binding domain. At the time, of course, the receptor binding domain was known to bind, and it was known to have a flexibility, but really that it was so much concerted with the N-terminal domain was not obvious. So we did that approach. We wanted to have something more powerful, and we got into deformation field estimations. Essentially here we said, listen, we are going to model the reality in the following manner. You have the macromolecule here. We are going to define a deformation field that will deform the structure of the macromolecule so that each image that I get corresponds to an average structure plus a particular deformation for that particular image. So we were going to estimate the conformational or the deformation field of each of the images. It was a big task. I mean, you have 100,000, 500,000, 1 million particles. We aim to estimate the deformation field. Well, you have to be very careful, because really there have been methods in the past that have tried to do that, normal mode analysis. Now there are other methods based in neural network. What were we going to offer of something new? Well, we thought about the following. Listen, we are not going to try to do too much. We are not going to try to estimate for each image too many things. What about if I express the deformation field, not the structure, but only the deformation field as a certain base, and I work to find the coefficients in that base? A very analytical approach. Well, that's exactly what we did. The base was a little bit complex, but we didn't have to find more than 30 or 40 numbers per image, because each number was to be multiplied by each of these elements of the base and would get not the image, but would get us the deformation field. So the deformation field was going to be calculated with few coefficients, not to get into what is called overfitting. Overfitting typically happens when you try to get too many data, too many parameters out of a lot of noise. Then there are many ways to fit that. Because your degrees of freedom are very large, you have a lot of noise, so you can do marvelous things. But normally you get to the wrong answer. So we did that. And how did we find this deformation field? And I promise I will try to put very few slides, very few mathematics or things like that, but it's a number of conceptual things. Really, what do you have? The single thing you have are your images, your experimental images. This is what you have. So everything you do has to take as reference your experimental images. So here I'm saying, I want to find the deformation field. The deformation field of what? Of an average structure. So I have the average structure, I apply a first guess of the deformation field, and I generate a computer simulated image. Well, the way I get JL, I don't really know, but I'm pretty sure that if I am wrong with this guessing, this computer simulated image is going to be very different from the experimental image. And if I somehow start changing this JL, it will happen that this computer simulated image will be very similar to that experimental image. And when this happens for 500,000 images, okay, I have found a good local minima for the deformation field. Okay, so this is what we did. And the way we obtain this JL, optimization approaches, then neural networks, these things. So this is what we get. This is a typical test case that is coming from one set of ribosomes with a drug. And the point is that each of these points actually is an image out of 200,000 or 300,000 images. And what we are seeing here is a two-dimensional representation of this 40-dimensional deformation field. And we don't take this just is a map to tell us if they are clustered or if it is a continuum. And here we see that it's quite continuous. We see we can take different samples at different parts of the conformational map, and we do see that there are changes in different parts of the conformational. So we are not taking the images and making one average 3D reconstruction. We are not taking the images and making two classes because I have no idea how can I divide this into two classes. What I'm saying is that I am able to walk through the map and give you a snapshot of what seems to be a continuous of conformations. Okay? Again, understand that I give you the snapshot, but I'm not able to order them temporally. Okay? This temporal order is chimera temporal, or chimera X temporal order. It's not, well, you can certainly, what I'm saying, you can explore this map and you can find really how everything changes, and with that you are exploring a new dimension that before was more difficult or impossible to explore in Cryo-EM. And an example that we use with HER2, a growth factor that is quite important in a number of cancer, especially breast cancer, and in complex with one of the therapeutic antibodies that are used for this cancer. So, and this is a two-dimensional map, two-dimensional representation of several 100,000 images of HER2 in complex with this trastuzumab. We don't see two classes. What we see is actually a continuum of classes. We can really take a snapshot of this continuum, and when we start analyzing this continuum, we see that there is an extended conformation and there is a continuous of changes until a more compact conformation. And really we can just take a few and we can see which are the differences without the therapeutic antibody, and we clearly see that something that is bound here is producing an allosteric change that has a reflect here in the upper part. And that just happened that this part is the one responsible for dimerization of HER2 with HER3. So what we are saying is that this therapeutic antibody, one of the mechanisms of actions is by blocking the capability to make dimers of this. And really, when we substitute it into other structure in which we have dimers of HER2, HER3, we see that simply, I mean, the extended form is very good to receive the arm of the HER3. The compact form is incapable of receiving any arm from the HER3. So one of the probably many ways to work of the therapeutic antibodies is this one. These are just coming result on the spike of COV2, a number of variants. Here I am presenting a very initial one, the D614G, and the one of the Omicrons, the BA2. And the first thing, the images have been processed in parallel, taken in the same microscope, everything, and you see that looking at the conformational space, well, there are two clouds. Clearly, the variability that the Omicron has nothing to do with the flexibility that the D16G has. Completely different. And now we can zoom. Zoom means take these images and make a separate analysis. And we get really the typical conformations of the spike up and down and all that before you had to go through classification. Now you get them immediately by inspecting the conformational map. And you see how the variability of Omicron really is much larger, but still we see a pattern that we are exploring at this moment, really. And this is the power of these new methods. Let me tell you now, I hope to have convinced you that we are able to take 100,000 images or 500,000 images, and for each of them have an estimation of really how much was deformed the average structure to produce that particular image. And now I go to something else. Reconstruction algorithms that are at the heart of everything we do and at the heart of what the X-ray scanners are and all that, assume that you have projections of the same structure. If you have projection, but each time is a little different, the structure, because the flexibility map is different, when you do a reconstruction from this set of images, you get a reconstruction artifact, because the algorithm does not know anything about making a map out of a set of images which are not exactly projections of the map. So we are going to change the reconstruction algorithm so that we integrate the model that the images are not all of them coming from the same average structure. They are coming from a deformed average and deformed in a separate manner for each of the images. So we are not going to do any unreasonable thing of going back from flexibility. What we are going is to take into account flexibility correctly into the mathematics of 3D reconstruction, nothing more. So this is what intend to see, is that you can really explore the landscape of solution, or you can try to bring many elements of the landscape of solution to one single element and increase the resolution in that manner. This increment resolution is what I intend to show with this. But better to see. How we do it? Well, it's really very simple, because we are using a method that was invented, if you want, in the 70s, is arithmetic reconstruction technique. And here we are making, this is the formula for the method, and here we are incorporating the deformation field, mathematically very small change that normally was impossible to do, because this would have, this 10 years ago would have taken ages to do it, okay? The difference is that now many of these methods can be mapped into GPUs, and you forget. It's 10 times faster, or 10 times, 100 times faster. 10 times is that you have done a very bad coding into GPU. So now things, can work in a couple of hours. You have everything done. And indeed, let me go to this. This is a zoom of the ATS ribosome dataset that I was showing you, and you clearly see how elements that were not so well resolved before now are perfectly resolved. And we can do one more thing, is I have told you that we have the reference map, we have the Cernic 3D to estimate the deformation field, and then we have the 3D reconstruction, okay? What about if we do it iteratively? Once you have the reconstruction, you again do the alignment, do the Cernic, do the third. Well, for those biological systems in which the model really applies, in which you have deformation from a reference structure, the results are amazing. I have taken a slide away because there are a number of systems that is being applied, but the results are not yet to be published, so I'm not showing the maps, but I'm showing something that is the quantitative estimation of the improvement. I am focusing here, what you see is I am focusing into the most complicated part of the map, and I am calculating the local resolution. I am calculating an histogram of the local resolution. And this is the part of the map that was more blurred. The local resolution was more or less 6 Armstrong, more or less. And then when we go to SART, deep Cernic 3D SART, we get an improvement in local resolution that is a lot. So really now we are able to see the structure much better, because before we were just mixing together images that were not coming from exactly the same conformation. Here, with all the errors in estimating the parameters of the flexibility, still we are doing much better than without making any estimation at all. And of course, these things are sort of complex. We understand that. So in our software, we have created an environment that is called Flexibility Hub. It has just been published in ACTRIS-D. And what we are doing in that environment is to offer you a number of different methods to run, and a way to try to understand the result of these different methods at the same time. Of course, I am just finishing. I have started from the very first moment that we are limited by many things, among them that we don't have temporal information. Temporal information, we think, may be coming only in two manners. One is that you design an experiment or a set of experiments in which there is time information experimentally, because it's time zero plus 100 milliseconds, which is our order of magnitude doing things. Time 0 plus 200 milliseconds, something like that. So there is a time course, perfect. And the other thing is that molecular dynamics could give us also the time dimension, now that we have a whole variety, a whole ensemble of maps that could guide molecular dynamics. This is starting. We are collaborating with a number of people in the field. It's not that we have anything clear, but I am convinced that we are at the right moment to make the connection cryo-EM molecular dynamics in very concrete manners. Something that we have done is in our environment, Scipion, it is also now encoded all the PRODI environment. PRODI is one of the, here is Yvette Bahar software, so the two of them interconnect very well. And this has been used already when we were studying something of a particular variant of the spike, the A22V. Probably you have never heard of the A22V mutation, but in Spain it was very important. So when we studied that mutation, we found this is molecular dynamics, starting from maps obtained from cryo-EM. We found that really the open state had a different dynamics than the dynamics of the open state of D614G and of the Spanish mutation really was different in a defined manner. we keep doing things like that. So in the nutshell, we obtain a quantitative information on macromolecular continuous flexibility. We computational undo deformations. Undo deformation means that we take into account correctly the 3D reconstruction algorithm, that we have integrated everything into an environment, and this is offered as an Instruct technology. Instruct is the European Research Consortium for Structural Biology. We have had a workshop on, perhaps the first workshop, on flexibility, on cryo flexibility about 3 weeks ago. We have a calendar of courses every year, and first quarter is single particle analysis, second quarter is going to be flexibility, third quarter is 4 facilities, all the first step of the pre-processing pipelines, and the fourth in December normally is tomography. I'm just telling you in case you want at some time. They were all online. Now we are changing into physical things. But you may access that through Instruct and through this project that Kildare was also talking about. So this is what I'm doing in the nutshell, and the group is this one. We have good funding in spite of the difficult times. The Cerniquez idea and all that was, or is being done by a very talented PhD student, David Herreros. The connection with molecular dynamics is mostly, not only, but by British postdoc, James Krieger. And here we have Carlos Oscar Sanchez-Orzano, that is the co-PI in the lab, and he was my pre-doc, postdoc, and now we work together very well. So I am open to questions, and anything that you find interesting, just let me know. Thank you.
Questions from the audience? Fabio.
Thank you, Jose. It's not clear to me how do you compute the deformation fields? How do I mean? Do you use the normal mode oscillations and to compute the deformation function, the GL function? So you are assuming the normal mode oscillations to do that, or you don't assume anything?
At this moment, we don't assume anything. I tell you, perhaps. Okay, how we did it 1 year ago was we put all that into a certain optimization algorithm that is called Powell optimization. It's a local optimization. Is not exactly a gradient, optimizing the gradient, but is similar. So you go in the direction in which you expect to have locally more changes, and you stop in a certain local minima. How we do it now? We do it with a neural network. So we present the images to an autoencoder. There is a latent space, and now the decoder, instead of being trained, is the physics. We say that the coefficient of the latent space, we don't know if they are Cerniquez, but they better be Cerniquez, because we are going to treat them with the mathematics of Cerniquez. So, and this is how we do things right now. The latent space is taken from the results of an autoencoder neural network, and is much faster than any other thing we have done.
Okay, very nice talk. Thank you. I was wondering also the same, almost the same, because you have only projections in 2D. How to choose the right principal components? This is the main question, I think. And how you do the mean, this average.
when you do optimization, something that is critical, because you are going to do always local optimizations, okay? There is a way that is very well known to do global optimization, that is to check all possible solutions, okay? That is simply not possible. So since you are going to be stuck into local optimum, you better start not too far away from the optimum. So what do we do here? We take the images and we either work with a certain class, so we follow the discrete approximation and we say, okay, class 1. We focus into finding the variability in class 1. Or we take the whole set of images. In the case of the ribosome goes like that. In the case of that example that I have only shown you the local resolution histograms, we have taken the complete set of particles and we have assigned with Relion or with CryoSpark, which are the two main programs. So the two of them using maximum likelihood expectation maximization algorithm, we have assigned the pose, the geometry, and with the pose of each image, with the geometry, with the alignment, as we say here, we have done a normal reconstruction by back projection. We have calculated, this is the way in which we have calculated this medium resolution map.
Thank you for your talk. My question is, could this analytical approach be useful to improve the analysis of sample with a large number of conformations, for example, denatured protein?
We consider that this approach and these types of approaches are those that can give you a richer set of answers, and they map better to the reality of your data. A way to answer you that I think is not exactly what you're asking, but is for the general thing perhaps, is that we have asked ourselves many times, are we in good biophysics terms? Can we make studies of free energy and we think that we still have a lot of variability in these maps. What we are doing is to get consensus of different approaches using, to get the consensus, another neural network. And once we feel that we are happy to go that way, we might get into the biophysics. We are also, I mean, the time domain we need very much with molecular dynamics. And if we do coarse grain, we forget the time dimension. So we have to go to atomistic. But we are there. We are there.
Paulo? You have said that you are going to the molecular dynamics, but why you don't have this option at the beginning? Was very difficult to make even some approximation. I don't know why. Because for me, it's the only way to see clearly that, but maybe it's my opinion.
Yeah, I mean, the point of molecular dynamics has always been on the table for already some years. I am trying to remember the name. Joachim Frank has collaborated for many years. A very well-known German scientist that died several years ago. Well, but the point of finding at that time was the modeling of the structure of the ribosome to the low resolution maps. This is another problem, and there they had all the issues of the data, the number of atoms, and these things. We didn't go for the. I mean, here we have to get the pose, the orientation. This is the big thing. And we are not refining for that, but we could. I mean, this is the next thing. Is instead of 40 parameters of the deformation field, 45. And these 5 are a recalculation of the orientation projection. And this would be probably the way to gain a little bit. But with molecular dynamics, what we try to find is your molecular dynamics supported by our experimental data? Which trajectories are better supported? What do we see? And there, I think it's a type of question that this is what we are doing right now. Is strictly seeing if we can provide support for simulation, more support for some than for others.
So this is something that we, from time to time, we come across, which is validation, right? So this is a big thing either for cellular tomography or subtomography averaging. So what is your vision about that? Because this is something that sometimes is worrisome because everybody is doing a lot of things with softwares, and how do we validate the data?
The validation is critical, and here the best validation method that we have is once you have the resolution to get to do a mapping or to do a structure modeling, and then you try to say how is the model in agreement with the map, and that is one way to validation. I think is the most powerful way to validation. Of course, this assumes that you have a map of a quality high enough to make a tracing. Otherwise, it's not. And if you don't have that, you are left with a lot of densities that you don't know what to do. Once you assign these densities or think that you are assigning those densities to chemical entities is when you can make the question, is this assignment correct? Before you have clouds of density points.
Very nice talk. So I have a suggestion. I don't know if you, if I got your point. So part of a blurry that you can experience in your 2D data, it's based on a lower or low populated states of the protein. My point is, have you guys ever tried to populate these lower populated structures and try to make these snapshots just managing these lower populated states and sum all of these data sets to improve these lower states in your final data set?
We are telling you that each of these points is an image, and each of these points is the projection of the average structure plus a linear combination of 40 elements of a base. So from each point, I can obtain a 3D structure. However, one has to understand also the limitations. Your projections, by definition, are only experimentally giving you information in a certain region of the space. If you go to Fourier space, it's very clear. In other, they are not providing you information. So why really can we go to give you a 3D from one point? Well, because the base is designed in such a way that you have 3D functions. So, of course, if I put a number before this function, I give you a 3D thing by definition, because the base is already 3D. But which is the confidence I have that that point is really has the information to give you a 3D structure? Well, I know that experimentally I do not have that information. What should I do? Well, I could take a number of images here to make a reconstruction out of these images, and all that. The problem is that you have a limited number of images. So for that, what we are sort of doing is increasing. For it, you want me to give you the structure of this, which is the 3D structure that gave rise to that particular image. Boof. Okay? I take a radius of these images, where I know that I am mixing things, but since I know that I have an estimation of the deformation of each of these images and a way to correct for that, I give a 3D structure that takes all this information into account. Is what I try to represent here. So this image, you want me to get the 3D map out of this image. It's complex. What about if I try to get them for the rest of the images at the same time by compensating for the deformation that I have calculated? So this is what we are doing, and when I present you this data, this is what we do.
A question regarding the deformation map again. When you compute the deformation method, you used a medium resolution map. And how is the capacity of predicting the GL function depends on the resolution? For instance, if you have a low resolution map in the beginning, can you be biased by the local resolution, a low local resolution in some part of the protein, and infer some flexibility in a region that is actually not flexible, something like that?
All these methods have also the following limitation. We work normally in 128 by 128, so we do a downsampling. We are smoother. We try not to have too much noise to estimate. So this, which is the bad part of what I'm saying. I'm saying that you are not going to reach high, very high resolution if you are by definition doing a downsampling to start with. So the way to get to high resolution is first to correct for these things, and then to directly go into another round of alignment, refinement, and all that. Hopefully taking into account the deformation parameters. But go back to the images again.
So I have a last question, more philosophical question. So we see that crystallographers, people from NMR, Cryo-EM, molecular modeling are working together now. So how's how do you think that this is going to progress? How each field can contribute to each other? Because we have different backgrounds, right? But we are seeing like Fabio, which is strictly an NMR guy, which is moving to Cryo-EM. So how do you think that this will, the contribution of each field will make a difference?
Yeah, by definition is the best way to go to a global or more global optima. So for many years, Cryo-EM has been a field that was not closed, but more or less closed, because we were reaching only blobs information. We were a marginal technique in structural biology. It is now that we are not, but then something that, for instance, was completely lacking in the EM field, and we had, and we still have, this is a personal opinion, but less training than the crystallographer is in modeling. I mean, we have much less of a feeling of atomic structures than somebody who has worked only with atomic structure. We have a feeling of the blobs and the density modification and the thing, while this. So that's something that, for instance, Alejandro Buschiazzo from Pastel Montevideo, was last year with us in Madrid, and it was clear. He was light years better than us in terms of modeling and understanding. And in terms of interactions, is critical. And however, or time, anything related to dynamics, even if dynamic depends a lot on which time scale we are talking about. But we don't have a feeling of dynamics, and NMR has a feeling of dynamics. So I hope that this would be the way to get better than being alone.
Okay, so if we don't have any other questions, so we thank again Professor Carazo. A round of applause.